we're called mitogens. I know this is like hair in the background. And mitogens will activate cyclins, and you will enter the cycle and divide. And then we talked about how there were checkpoints in your cycle. And in these checkpoints, one of the things you would do is figure out if the DNA was correct. And that was done by, remember P53? That's also going to come back with cancer. So it's kind of the one gene that is really important for your ability to check that the DNA is correct. And if there were mutations, then the cells would be sent to apoptosis. The cells would be sent to cell death if you couldn't fix them. So what we're going to be looking at today is actually how do we make cells die. So the first question is, can you think of different times in your life where cell death is a good thing? I just gave you an example. There's a lot of damage to the DNA. It's a good thing to be able to kill the cell. Can you think of any other times when we may want to kill cells? Eh? So the hair in itself is protein, but the cells that give origin to that, they could die. Depending on where the hair is, that could be a plus or a minus. I would think, yes? So during development, you know that we all look like frogs during development. We have these awesome webbed fingers. And then those are all cells that need to die. And if they don't, when you're born, the doctors cut that out. You don't remember it. Yes? So when you're in your nose, we talk about how cells die because they get damaged by the chemicals or by you blowing your nose. So we want to be able to kill the cells that are damaged so we can replace them. Anything else? Yes? When cells are infected with viruses as part of your immune response, we actually kill cells. So all of these forms of killing are very, very organized. Apoptosis, the other name for apoptosis is programmed cell death. It's a set of steps that cells take in order to die in a very organized manner. So this is the example of the webbing in a mouse hand. So you can see all of the cells here that will eventually die so you can have actual fingers. It's cute. There's two ways cells can die. One is called apoptosis. The other one is called necrosis. My personal analogy is apoptosis is when you get to the end of your life and you made a will and you have your trust and you invited your family over and they all come to spend the last couple of days with you and you're happy and at peace in your life, in your bed, and that's like all beautiful. When you die, your family doesn't have to worry about it, everything has been paid for, funeral arrangements are made, you are a fantastic person in that context. Necrosis is when you actually just have a car accident and everything explodes. And you're not ready for it, and like it becomes a whole mess and a nightmare for your family. So if you put yourself as a cell in your body and your family is the rest of your body, apoptosis is when a cell dies in a very organized manner. So the cell has a problem, looks around, starts breaking its DNA, starts opening its mitochondria, breaks itself down into wonderful little vesicles with pieces of itself, doesn't bother anybody around it. Those little vesicles get eaten around, nobody even finds out the cell died. It's a very, very organized way of dying. Necrosis is usually the result of a lesion. If you get like a bruise, if somebody hits you, and that usually means the cell just explodes. There's no plan, there's no organization, and everything that's inside of the cell spills out. So your lysosomes that have all these enzymes that can degrade things, now they are exploded and you're releasing those enzymes to the environment. So the cells around you are going to be damaged. It usually doesn't involve just one cell, it's a bunch of cells all dying at the same time. Because you're releasing a ton of things inside of the cells, it usually causes an inflammatory response. So then that area gets red and it gets painful, and it's kind of like this mess. Does that make sense? Like the distinction between apoptosis, like a really nice, organized way of dying. So this is kind of like a comparison of that. Necrosis, the membranes break. So if the cell membranes break, water will come in. And substances from the membrane broken down into pieces is very different. In apoptosis is a very organized way of breaking DNA. That's called ladder-like. And while in the process, you're just going to break DNA randomly. So this is trying to show the differences. This is a normal cell. This would be necrosis. You see the exploding part of it. While in apoptosis, the cell changes, makes little pieces of itself. That's called bleeding. And then these little pieces get eaten by other cells, and that's fantastic. Yes?
You said that with necrosis, it usually affects large areas.
Yeah.
If the cell membrane is breaking and all the water and fluids and parts are leaking out, how come, let's say, if you get necrosis, like an arm, it doesn't just turn into a mushy mess?
So when you first, imagine that you burn, or you just, somebody hits you something there. Initially it's going to swell, you're going to get blisters, or you're going to get all this water, and then it's going to dry. Because once you lose the kind of like cover of the skin, let's say, now water can escape, and what you get is a very dry area, but that's mostly because you lost the permeability of the skin. And but the contents of the cells usually do spill out, and the initial thing you get is like a mixture of blood, liquid blood, and swelling. Yes? During apoptosis, inside of the cell, the organs are destroyed, they're not recycled. Anything else? This is another cartoon version of the same idea. Necrosis, normal cell, swells, eventually breaks down, blah, blah, blah. Apoptotic cell breaks into tiny little pieces and blending with intact membrane, and then they get eaten by other cells. So there's really no mechanism to study necrosis. It's like you have an accident and stuff explodes. There's no sequence of events. There's nothing really except like it's a mess. So we're going to focus on looking at apoptosis, and we're going to look at it more conceptually. There's very specific pathways where protein A activates protein B, which activates protein C, etc. We're not going to go into that level. We're going to kind of stay a little bit of a higher, because you guys have already a lot of things to keep in mind. So there's cool things about your body in development. We actually, during development, we have a tail. They guys would love to have a tail. I thought about that, it would be so cool. You actually have a tail when you're a baby, and then the tail goes away. It goes away through apoptosis. It's a very organized thing. When we are babies, we have both female and male genitalia. And then during development, one of those gets selected and the other one disappears. It disappears through apoptosis. So all of those cells undergo programs of death. Oops, sorry. We talked about webbing when we move the tissues between the hands. Because you have to think that the hand starts as a little ball, and then you start kind of making fingers in that ball. It's super cool. If you look at your blood vessels, that's the other. I love that. Blood vessels are initially solid. When you make your blood vessels early in development, they're totally solid structures. There's no space in them. All of the inside dies by apoptosis, so they become hollow tubes. Without apoptosis, you would not have a circulatory system. Programmed cell death, or by the way, apoptosis has the most pronunciations of any word I've used in science. So you can pronounce it any way, it's going to be okay. People say apoptosis, 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 any way you want. It's fascinating. English language. There's two ways you can induce this very, very well-planned program of death. One is called the suicide pathway, and the other one's called the murder pathway. So the murder pathway is when your body wants to kill a specific cell and you send a signal that triggers the molecular pathway that leads to cell death. So for example, I think these guys are supposed to be some form of like some damage to the cell. I have no idea what's going on here. If you have a cell infected by a virus, your immune system will actually signal that cell to die. It will murder it. It's not the cell looking, oh, I have a virus, maybe I should die. No, actually one of the things viruses do is they interfere with your apoptotic pathways. So that when the immune system tells the cells to die, like the virus goes like, nah, 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 and the cell doesn't die. It really does a dance there, you can see it. So they block the pathways. Suicide is when what that happens with p53. So that's a cell looking at itself going, my genome is not correct, I can't fix it, I should probably die. And the molecular components involved are actually different. The easiest way for a cell to die is if you don't have trophic factors or growth factors present. So when we talked about the cell cycle, we had a list of factors. One of them was mitogens, which induce cells to divide. But then we have growth factors, which keep cells alive and make them grow. Most of the cells in your body will die without growth factors. So I take cells from any part of my body and I put them on a dish, they die. They actually need specific chemicals to keep them alive. And one of the things those chemicals do is they're constantly preventing apoptosis. So growth factors promote cell development, promote making new proteins, but a lot of them also prevent apoptosis. The one cell type that we have that does not need growth factors or trophic factors are cancer cells. Cancer cells can keep themselves alive happily. So the steps of apoptosis are like, the cell starts to shrink, the chromatin is going to start to condense, then you break the nucleus into pieces and you get what's called blebbing, from the little blebs. And then the blebs get eaten by phagocytic cells, and usually the thing that gets eaten is called an apoptotic body. Yes? Nothing, in reality. The only thing is that growth factor could be released from one cell to the one beside it, and if it doesn't go through the blood, then you don't call it a hormone. But the effect is the same. It's just how does it get to the cell. Are we okay? This is the list of steps that are involved in apoptosis. So for a cell to die, you have to induce apoptosis, and the first thing you're going to do is you're going to damage the mitochondria. When you take biochem, those of you who have not, you'll see that one of the awesome things that we do in our mitochondria is the electron transport chain. You guys remember from initially in the class that you make ATP, you have the electron transport chain in the mitochondria across the inner membrane. There's a gradient of protons across that membrane. As the protons go through, you can use that energy to make ATP. And there were different proteins involved in that. One of those proteins is called cytochrome C. So, for a cell to die, cytochrome C has to leave the mitochondria, which is a bizarre thing. It suddenly has to exit from where it was doing and go into the cytosol and interact with other proteins inside of the cell. When people first proposed this, the scientific community pretty much laughed. There was no way that the cytochrome C was necessary for apoptosis. This is a protein involved in the electron transport chain. Its job is to take an electron and pass it on. That's its life. And the people working on this actually had an incredibly hard time publishing that cytochrome C was important for apoptosis. They kept saying, well, we find it in apoptotic cells, and people would be like, well, that's because you're damaging the cells and you damage the mitochondria. And so they had to actually be a very big push from specific members in the community to get this approved and checked, because it was so abnormal. Cytochrome C binds a protein called APAF1. We're not going to worry too much about this, but it is important that it has to leave the mitochondria and it will go to the cytosol. If your proteins are leaving the mitochondria, you're actually going to be damaging its membrane. So the potential across the membrane is slowly going to disappear. You activate a set of enzymes called caspases. Have you ever heard of caspases? Yes? No? Caspases were identified as the key enzymes involved in apoptosis. We have about 13 of them, and we're not going to see the pathway, but the pathway is something like caspase 9 activates activates caspase 5, which activates caspase 3, which then activates another caspase. So the goal right now is to know there's a set of enzymes called caspases that are involved in this process. And then the next steps, one of them we talked about before, we talked about membranes. We're going to come back to membranes for a moment. The exposure of phosphatidylserine. Phosphatidylserine is normally an intracellular, it's on the internal leaflet. And during apoptosis, you will put it on the external leaflet. That is what allows this guy to know that he can eat this thing. The fact that the phosphatidylserine is on the outside. We're going to review that in a moment. There's changes to the cell, and then we break down the DNA. And this whole thing will take about 2 hours for a cell. There's a way you can study these different steps. So this is a cell. If you can see, there's a border here of the cell, and then there's like a black thing in the middle. This black thing is the nucleus. And then this is the border of the cell. And then there's these white dots everywhere. Are we okay with that? What we're looking at? It's a cell with white dots and a black nucleus. Here's another cell, here's another cell with a black nucleus. The shiny dots are cytochrome C that are concentrated in the membrane of mitochondria. They look shiny and bright because they're concentrated. On this side, we're looking at the same type of cell, but that is undergoing apoptosis. So now instead of having this very bright signs of cytochrome C that you can see in specific locations, you have kind of like a gray pattern because the cytochrome C has left the mitochondria and is now everywhere. So it's not concentrated in any one spot, so you kind of get this like light staining. Does that make sense? Here, cytochrome C is localized in mitochondria, so you get, when you stain it or when you put an antibody against it, you get a lot of shininess wherever they are. So it would be like there's probably a mitochondria here, etc. When a cell undergoes apoptosis, this pattern changes completely, and now cytochrome C is not localized. You find it everywhere in the cell. So if you're looking at cells and you stain for cytochrome C, something like this is a clear sign of apoptosis compared to something like this. Yes?
How does the mitochondria never release the cytochrome C?
So how does the mitochondria know to release the cytochrome C? There are receptors in the membrane that if you activate them, will go and activate a signaling pathway to phosphorylate targets, including cytochrome C, and it's somehow that triggers the being released. So you need a signal to do it. Yes? Yeah, in the inner mitochondrial membrane. Yeah, so it's going to have to detach itself from the inner mitochondrial membrane, which is pretty hard to do, go through the pores of the outer mitochondrial membrane, and then hang out in the cytosol. Any other questions? So the other thing I talked about was phosphatidylserine exposure. This should look like a familiar graph. We talked about the different kinds of phospholipids that we have in cells. We talked about how in the cell membrane we could have an isodistance for something else, sphingomyelin, phosphatidylcholine. These two tended to make cells more membranes more rigid. Then we talked about how phosphatidylserine and ethanolamine were mostly on the cytosolic side of the membrane. So this is the external side, this is the cytosolic side. And then we talked about how cholesterol was equally distributed. So there was this idea that the cell membrane has different kinds of phospholipids, and there's an asymmetric distribution of them. There's some of them are more on the outside, some of them are more on the inside. In a healthy cell, phosphatidylserine is only on the inside. It's pretty much on the inside only. Excuse the cartoon, for some reason there's not a lot of good pictures of this. So this would be the normal cell. But an apoptotic cell will flip the phosphatidylserine to the other side. And we talked about the different movement that lipids could make in the membrane. We talked about how they could rotate, they could move laterally, and all of that was easy to do. The one thing that was very hard to do was to flip. Spontaneously, it pretty much never happens. We actually need enzymes that can move one phospholipid to the other side. Remember how they were called? Fleet bases. So I mentioned there's fleet bases, flow bases, depending on which direction you're going. But in general, we're just going to call them fleet bases. So there's actually enzymes that are recruited to put phosphatidylserine on the outside. So you can see it's a really well-organized process. It's not by chance at all. It's not like exploding itself. And this phosphatidylserine on the outside is what phagocytes, cells that eat other things, recognize in order to eat this cell. And then the last hallmark of apoptosis, so this would be the signs that you're looking for to figure out if a cell is undergoing apoptosis or not. The last sign is what happens to the DNA. So DNA wraps itself around histones. We talked about how DNA around histones is called chromatin. And chromatin can be more or less compacted. So it can be heterochromatin or euchromatin, and that affects the expression of genes. If we look at how actually chromatin is made, the loop that wraps itself around the histones, so this kind of thing here, so like DNA around the histone and coming out, involves 180 base pairs. So that's kind of the magic number. When a cell dies through apoptosis, one of the things that happen is that chromatin becomes loose. And enzymes come and start cutting in between the histones. So they're cutting. So if they could get all of them, you would see all of the DNA as 180. Usually they skip some, so you get 180, 360 if you have 2 of them, 520 if there were 3 of them. Does that make sense? It tries to break it down as much as possible, but it's not a perfect system. So you get either 1 or 2 or 3. When you have the DNA wrapped around a group of histones, that's usually called a nucleosome. So we get 1 or 2 or 3. So it's either going to be 180 or 360 or 520 or 700, depending on how many of these you have together. The enzyme that cuts this is not important, but it's called an endonuclease. Endo means inside, nuclease means I break nucleotides. So if you look at the DNA that comes from a cell that has undergone apoptosis, so I'm going to take a moment. This is a gel. The way that this works is you put DNA at the top, and then you run electrical current and you wait. And the molecules of DNA are usually charged. What is the charge of DNA? Negative. So they're going to move in the electrical field. And the charge is proportional to how big the DNA is. If you have 20 nucleotides, you'll have 20 phosphates, you'll have 20 negative charges. If you have 150 nucleotides, you'll have 150 phosphates, you have 150 negative charges. When you do this kind of gel, where you put DNA and it runs according to charge so you can see it separate, this is called, how does this called? Any ideas? It's called a southern blot. S-O-U-T-H-E-R-N, like south, but going southern to the south. The reason it's called a southern blot is because Dr. Southern invented it. It has nothing to do with going to the south. The guy's last name was Southern. So it's called a southern blot, S-O-U-T-H-E-R-N. It's a DNA gel. When you take genetics and biochem, you'll see that there's other gels. So if you're running RNA, it's called the northern blot. And there was no Dr. Northern, it's just that whoever thought, oh look, one is called the southern blot, so we should call the other one the northern blot. And then if you're running proteins, it's called a western blot. So DNA is southern, RNA is northern, protein is western, and there's no eastern blot. Just so you know. You may invent it, according to size. And there's 3 lanes here. So you put DNA here and it ran. The first lane says marker. What this means is whenever you do a DNA gel, raise your hand if you've ever run a DNA gel. Woo! Look at that. About half the class has run DNA gels. Was it fun? Yeah, the first time is fun. When you do it every day, it stops being fun. Like I remember the first time I ever ran a gel, I was like so excited. I had it on my hands and I was gonna take it to the UV machine. And I was like walking slowly with my first gel ever in a lab, and I dropped it. So I then picked up the pieces of my gel and put it into the UV machine anyway. Because I was like, it's my first gel. So, and after that I did it so many times I don't care anymore, but I was very excited about that. So the first lane, the marker, you use it as a way of, you buy a product that gives you pieces of DNA of different sizes. So you'll know that this is 100 base pairs or 200 base pairs. This is just to help you know the size of the DNA. This is here the DNA of cells that you did not induce apoptosis in. Number 3 is the DNA of cells that you did induce apoptosis in. So you get one bond, the other bond, and they're spaced very organized. So this is 180, 360, 520, 700, etc. Does that make sense, what you're looking at? If it was a necrotic cell, everything would just look like this area. You just have DNA of every possible size. There's no pattern. It would just be the whole thing would look like spreading of DNA. So if you're doing an experiment and you're trying to figure out if your cells died through apoptosis or necrosis, the easiest way is to extract DNA and run it on a gel. If you get a nice lather pattern, it's apoptosis. If you just get like, that's usually necrosis. Okay? Questions? Good? So we've talked about growth factors and how they keep cells alive. The question is, how do we know they even exist? So how did people discover there were things such as growth factors? And this is where development is really fun. Particularly in the 1920s, development was really fun. 1920s to 1970s, before the advent of animal protocols, what people did with development was very interesting. Some of this would be harder to do today. Not impossible, but harder. So they were experiments where people would take the eyes out of frogs, turn them around, put them back in. Or they would like add an eye to a frog or take eyes out. And like you'd see all of the what the changes that happen. So this is not in a frog, this is actually in a chick embryo. So these are all experiments done by a researcher called Victor Hamburger. Yes, like a hamburger. So these are known as the Hamburger's experiments. What he noticed, and this was the 1920s, and this awesomely looking creature, which looks like something out of like Ridley Scott's Alien movie, is a developing chick embryo. This is the spinal cord. And what he noticed was that if he took a section of the spinal cord in a developing animal, and he counted the number of motor neurons in the spinal cord, there would be a certain amount, shown here in red and green. But if he waited for the animal to be born, and then cut the spinal cord, and counted the number of cells, there would actually only be about 50% of them left. So during development, about 50% of the motor neurons in the spinal cord die. So that's initially the motor neurons are in a higher number and then they die. So of course what you would do is, well, you're wondering why, yes?
Related to talk with Dr. Gomez, how you don't have primary separate differentiation of muscle cells, you drop the number of motor neurons.
So it is partly related to that. This is a little bit earlier in that context. Because in that part of it, it's not that the motor neurons die, it's that they pull out from innervating the muscle. So here we're actually having half the motor neuron side. So he wondered why. And the hypothesis he came up with was, well, maybe it's just because they don't find a target. They don't find a muscle to synapse on. So how do I prove that? Let's go ahead and take a limb out. So this is called the limb bud. This would have become a little like cheap leg. So if you take this out early in development, initially you have a lot of cells, but after you take it out, the side of the body that lost the limb had almost no cells left. So it seems that if those cells didn't have something to attach to, they died. So how do you kind of like, remember we talked about take it out, see if it's sufficient, necessary. You take something out, you add it in. So he then added a leg. You see, you like fun experiments. Hard things to do today. You add a leg, and now what you see is that on the side where you have the extra leg, a large number of cells survive. So again, we're looking at what happens during development. The question is, why would cells die? You know that in most of us, the number of cells in your body before you're born is higher than the number of cells in your body by the time you're born. Your brain has more neurons than the ones you're born with. And this is a good thing. People have done in C. elegans and Drosophila where they have not allowed the neurons to die, so that all of the neurons that develop during development are there when you're born, and those animals do not function well. So through your development, a lot of the cells die, and in this case, how many dies seems to depend on how many muscles you could attach to. So this is kind of the summary. 50% of the original pool of motor neurons dies, and 50 stays alive. If you remove a limb, then you get less than 10% of the cells. If you add another limb, then maybe 75% of the cells survive. People usually ask, well, how many would survive if you add an additional limb? You didn't go for like 4 or 5 or 6. I don't know how many legs you need before to stop any of them from dying. So what he proposed was that the muscle was binding to the muscle, synapsing onto a muscle was doing something to keep those cells alive. He didn't know what it was, but he proposed that maybe releasing something that was keeping those cells alive. Are we okay at this point? Questions about this? Take 1 minute to explain to each other the hamburger experiments. Talk to each other. Hamburger experiments. If you have any questions, raise your hand. Any questions? Yes.
Is this true only for chicks or
What?
Does this stay relevant for all types and organisms or?
So these are the motor neurons. You know, you can also see the decrease in the motor neuron pool in mice. So people have shown that. The more complex of a mammal you become, or an animal, sorry, the more complex of an animal you become, the hardest it is to do this kind of things to you. So you can add in development a limb to a developing chick, and they will be born with an extra leg. If you add it to a mouse, it usually doesn't work. So these same kind of experiments have been done mostly in chicks or in frogs. And amphibians are incredibly plastic. But once you get to higher mammals, we have a lot of restrictions on what we can actually do. Like I find it fascinating. can take an eye out of a frog and develop, well, after they're born, they're a tadpole, they're swimming around, you take the eye out, and you're like, hey, and you put it back in, and it reconnects, and it works. If you were to do that to a human baby, it does not reconnect. And if you do it to a hamster, it doesn't reconnect. People have tried this. Not with a human baby, as far as I know. But with a hamster, with a cat, it does not reconnect. Yes?
What are you using to attach another limb? What's the process?
I don't exactly know how he attached it, but at this stage of development, just taking the cells and putting them in the right place would be enough. They would be able to survive and grow. Like you can make a tadpole of 2 heads if you take the cells that are supposed to become a head when they haven't yet, and you put it on another tadpole. The location and the cells are pretty. Remember we talked about cells being competent, being determined. So these cells have already been determined to become a leg. They just need the nutrients to be able to do that. Yes.
When you say putting them together, is proximity enough, or do you?
Now they have to touch.
Well, yes, is just touching enough, or do you have to make a decision to make sure the neurons connect as well?
So you can make them connect. So you can. What you do is you open the skin so that the cells can actually go in, and then you would actually just put it there, and you would just wait. If they get oxygen and nutrients, they're going to grow, and depending on the animal, it can become a completely functional leg. So when they did this to the frog, the eye was totally functional. Or it might grow, but it's not going to be as easy to control. They did this with. Have you guys ever seen the flies that have the antennas, the legs in the head? So you can do that with like just moving genes around. This is kind of the same idea. You just take cells that are expressing specific genes and you put them in a different place. They're already determined to become leg cells. The only need is that they need nutrients. Yeah.
So just to follow up on that.
Yeah.
Is the reason it works, like with amphibians and reptiles and non-mammals, does it have something to do with them being able to regenerate a little better, kind of how geckos can lose a tail and grow it back?
Well, it's all linked. So like geckos can lose a tail and grow it back. We don't do that. And there's been, particularly in the context of like cutting your spinal cord and people who have lesions, it is very clear that your nervous system has been wired to be very, very inflexible. So if you cut your finger, it hangs out and then you put it back on, you'll reconnect. Your peripheral nervous system is very user-friendly, let's put it that way. Your central nervous system will do everything it can to prevent the neurons from reconnecting. It actually sends inhibiting factors. It does. The idea was that at some point we decided that we decided to sacrifice plasticity for like structure and order. So your brain is really plastic initially in your life, and then when you kind of settle on something, it becomes much more inflexible. So the assumption is that there's some advantage to being that way. Yes?
What if you add the limb but you use it? There's something
So the question is, what if you added the limb and they really didn't use it? Yes, it will still be neurons because this is early in development. If they didn't use it, what would happen after they're born, etc., the muscles could atrophy and the neurons could die. But you would expect them to be born because they're not using their legs a lot when they're in there either. But activity is an important factor in keeping all of the systems working. Yes? Yeah.
If a cell reaches maximum division in senses, will it be less likely to die by apoptosis? Because it's not depending on people?
So if a cell is, the question was, if a cell is senescent, it reaches maximum number of cell divisions, is it like P53 going to be harder to activate? P53 is present all the time. So it would notice damage to the DNA. And it's not the only way, it's just the most common way to induce apoptosis. So people proposed that there had to be something that had to be released. And the experiments that proved this were done by Rita Montalcini and Sten Cohen. Why do we care about the names? Because she was a woman, one of the first women to get a Nobel Prize. I would have said, yay!
Yeah.
You know, it was hard. 1920s to 1930s as a woman working on all of the research. So, what they're looking at is this. We only have like 3 slides to go. It's just, they have noticed that if you took cells and put them on a sensory neurons coming from the periphery to your spinal cord. So what they found was that when they put them in culture, these cells would survive. So there could be two ways you could do this. One is that you just don't undergo apoptosis at all, or that you're releasing something that is keeping you alive. So they asked, well, are these cells releasing something? So they collected the media, and they found that the media, with whatever the cells had released, would keep other cells alive. So there was something released for the cancer cells that was keeping them and any other cell they put there alive. And in their beautiful innocence of research, they thought it was going to be the only thing that kept cells, neurons alive. So they call it the nerve growth factor. We know today that there's hundreds of growth factors, and different cell types need different growth factors to stay alive. It's one of the challenges of taking stem cells and making them into specific tissues. So how do you prove it works? Well, you take normal dorsal root ganglia stuff, you put them in culture, and you'll see that they die. You put the normal cells, you put them in culture, and by in culture I mean you put them on a plate and stare at them, like with some nutrients. You added the nerve growth factor, and they survive. So this one protein was enough to keep those neurons alive. And initially they thought, well, you know, this is going to be the only thing, this is going to solve all of our problems. So then they went and they tried a different group of neurons. So they tried motor neurons. Kind of going back to the leg experiments and hamburger, like what was keeping those motor neurons alive? What was the muscle releasing that was keeping the motor neurons alive? And of course they thought it was MGF. So they put the motor neurons in culture and they die. They put them with MGF and then they die, which was very disappointing. Many, many years later, people will figure out what the motor neurons need to stay alive. And it's a combination of two factors that you don't have to worry about. But it's called the brain-derived neurotrophic factor, or BDNF. And it's called neurotrophin-3 or NT3. So what people had to do was look at the muscle, collect whatever the muscle releases, and start testing it. So motor neurons need these specific things to survive. Different types of cells need different growth factors. And this is kind of how you can test for that. This is our last slide. The DRG in a normal animal, this is a bunch of neurons, and these little thin lines are axons. When you add MGF, you get that. It's like, woohoo, explosion. But if you use other ganglia from the body that are not the dorsal root ganglia, then it doesn't actually work as well. So, kind of the take home is that there are growth factors that keep cells alive. The default of most cells is to undergo apoptosis. Apoptosis is a very well-regulated sequence of events that lead to cell death. And it's important that you know the sequence of events and how to recognize the ones that we've talked about.